For anybody who is interested in having a say in our strategic planning, we have our new goal to fulfill 50% of market demand by 2040. And the board and staff are going to be holding meetings in districts coming up in March and April to talk about ways to achieve the goal and to get ideas from you, our members, on ways we can support you and ways to grow the industry, and just in general to get reaction to the goal and get your help to strategically plan how we should achieve it. So please check the Ontario Sheep Farmers website for your local district meeting time in March and April, and we look forward to seeing you.
Now let's get into today's episode.
Hi, everyone. Welcome to Flock Talk. I'm Erin Morgan.
And I'm Walker Ball, and we work for the members of the Ontario Sheep Farmers.
Today on Flock Talk, we're sitting down with Dr. Emma Borkowski from the University of Guelph. And thank you very much for joining us, Emma.
My pleasure.
So, Emma, to get us started, you were our very first podcast episode, so it's super exciting to have you back. And we're looking forward to getting caught up on all the research you've done since the last time we talked. Can you get us started, though, by giving us a background on how you ended up doing research on parasites and, just giving us a little background on yourself for those who haven't heard our previous episode?
Sure thing. So I was the typical kind of little kid who always wanted to be a veterinarian from when I was very, very small. And while I was working away in my undergrad towards that goal, I took a really great course on parasites. And it was just really fascinating to hear about how parasites interact with their host animals and how they cause disease. And there's this constant kind of arms race between the parasite and the host to try and one-up each other effectively, to try and, on the host part, keep the parasites at bay, and the parasites to try and get around those mechanisms. So I thought that was really neat and started thinking about how might I incorporate that into my future career aspirations. And then while I was in vet school, I started to realize that I really did love kind of thinking about these mechanisms of how parasites, especially, but all kinds of different things cause disease in animals, and understanding the kind of complex interplay that happens in those dynamics, and realized that what I wanted to do was pursue some research. So I had a chat with Dr. Andrew Peregrine as I was coming up towards the end of vet school to see what, if anything, he had going on with his research, because he is really kind of the master of the parasites when it comes to vet school at the OVC. And he had a project coming up that was going to be looking at trying to adapt this new genetic test for parasite immunity in sheep in Canada that had been developed in New Zealand and never tried here before. And the more I kind of learned about this, the more I thought that would be something really neat. So he was happy to take me on as a student, thankfully, when I finished up, pending that the project got funded. And long story short, it got funded. And so I sort of dove headfirst into dealing with parasites of sheep. And I won't say I haven't looked back since, but I've certainly been trudging stoically onwards and sort of expanding outwards beyond sheep into other species as well, but very much kind of maintaining my core of work with the sheep industry.
We appreciate you.
So how has your research evolved since then, Emma?
Well, what I started with in my PhD was kind of really a pilot study. And the good news was that it did seem to work fairly well, this genetic test that we brought to Canada, even though we have very different kind of environmental and management situations in Canada than they do in New Zealand. And so really what I've been doing ever since then is trying to get a better understanding of how Canadian sheep really develop this antibody response that we're measuring with the test, so that we can be even better and more effective at trying to use this test to find the best animals and incorporate that into how we actually manage sheep on pasture here in Canada and keep their parasite levels as low as we can in a sustainable way, so that hopefully our drenches will stay working longer as a last resort rather than as a first resort. So I've really been delving now from that first question of does it even work in Canadian sheep to what things might change how it could work from one farm to another. Because so far we're really reliant on doing the test on all of the animals on a given farm to see who's best on that farm. we're not as effective at understanding, okay, if you have one farm that's selling breeding stock and they do this test, how does that compare to another farm that's selling breeding stock that's doing this test? So I'm now starting to look into things like, how does the parasite species themselves that you have on your farm affect the antibodies that your sheep are producing? How does the sex, the age, the breed, kind of intrinsic factors about your sheep affect the antibody levels that they're producing? Same sort of idea is like, you can't fairly compare growth rates in a ram lamb and a ewe lamb unless you put in some sort of penalty on the ram lamb because we know they're going to grow faster. Maybe ewes actually are better, which is what we focused on at making antibody levels. And so we have to kind of put some sort of modification in there. And then also I've kind of branched into looking at what actually is going on a cellular level, because sort of the dream, the pipe dream that everyone has is someday we can make some sort of like vaccine, some way we can directly stimulate the immunity, which would work beautifully hand in hand with breeding for animals that are just genetically better at making immunity, because then if we can make them make a response faster without even necessarily seeing the parasites, breed for animals that make a better response, perhaps we can even protect them already going out onto pasture and help drop those parasite burdens down even farther. But there's a lot of kind of stuff that we don't know, because this antigen that we're looking at specifically, the carbohydrate larval antigen, or CARLA, behaves very differently from what most vaccines are oriented towards. We're talking about something that's in the gut. It's not circulating through the bloodstream of the animal. It's a very different kind of compound versus what we usually make vaccines to, that makes it harder to get an immune response to stick. So we really haven't done a lot of research on sheep, or really anything in this type of marker, outside of humans and mice for now. So there's a lot of kind of big question marks in there that we're trying to understand of who's actually responding to this antigen and making the antibodies, and then can we do something to make them do a better job?
Wow, that's really out of the box thinking. Because I was raised in Zambia, and we were always raised to do parasite cleanses once a year, and then coming here is kind of not a thing. But it's just so fascinating to hear. I mean, because they're so smart, they're such smart critters. I think I'll call them a critter. That yeah, I can imagine be really challenging even just to get on top of it, because they're always adapting.
Absolutely. And doing a sort of a single year, like once a year targeted treatment on everybody, cleansing, like if you will. The problem we have with that in Canada is we think actually that is partly why we've had such a problem with drug resistance develop here, because especially with Haemonchus, right, the big bad, your barber pole worm, that's the one that bleeds animals dry. It's a tropical worm. It does not survive winter. So the only worms that survive winter in Canada are the worms that are inside the sheep. So if you're then bringing out your ewes in spring and you're treating them all at lambing, guess what's going out onto your pasture? 100% worms that have survived the treatment. And so you have massive amounts of resistance develop really, really quickly. So this is part of the problem that we have of why there's such an importance in kind of looking at parasite management in a sustainable way, which doesn't mean no treatment. It just means we need to make sure that we're saving that drench for the animals that specifically need it, because the more drench we use, the fewer parasites we leave behind that actually are susceptible to that drench.
Excellent. So on that topic about looking at sheep's ability to mount immune response when exposed to parasites, can you share some key findings from recent work?
Sure. So most of my sheep work personally is kind of ongoing, but I have still been collaborating with Dr. Peregrine and his latest PhD student, Dr. Brad DeWolf, because they've sort of taken the next step in looking at CARLA immune responses. I looked at one farm in my PhD. Brad looked at, I think, 18 different farms scattered across the province, just to make sure that what we saw in my work wasn't a fluke. And the long story short is it wasn't. What Brad has found is that you do have animals with higher antibody levels in their saliva that we're measuring have lower levels of parasitism. So if you pick ewe lambs in fall, measure their antibody levels, those ewe lambs that have higher antibody levels will shed fewer eggs the following summer, which then also means that not only are they protected, but so are their offspring. Brad also found something that was really interesting, which was that the lambs of ewes with higher antibody levels actually grew better. And that didn't matter relative to like when the lambs were turned out on pasture. This was in lambs that were actually on pasture with their dams all the time. Some of those lambs were born indoors and only turned out on pasture after a couple of months. And yet still those lambs did better, even with that big gap of time where they never really co-grazed with their dams on the same kind of parasite contaminated pastures. So it did look very promising on the sheep side of things. My own recent work has sort of been tilted more towards the goat side of things. They have the same parasites as the sheep industry. And so it's really looking at trying to band together the sort of smaller commodity industries in the province that have a common problem so that we can have strength in numbers. And so I was curious to see whether goats might have similar responses to what we found in sheep, where more antibody means less parasitism. Not surprisingly, it was much more complicated in the goats than it was in the sheep, but we did still find some interesting and useful things, some of which were actually in common with what we found with Brad's work in the sheep. So in both sheep and goats, once you start getting really, really high levels of Haemoncus specifically on your farm, Haemoncus seems to have less availability of that Carla marker. So if you have too much of it and not enough of your other nematode species, it tends to suppress that immune responsiveness a little bit. They just don't make as much antibody as they do when you have other species of nematodes on your farm making a larger proportion. So that's something that's really interesting that I'm going to want to untangle a bit with my future work, which does plan to have a look at, okay, exactly like what is the difference if we have the same number of parasite larvae with this Carla marker of different species of nematodes, just how much of an adjustment might we need to make for a farm that's got, say, 70 to 80% Haemoncus burden on that farm versus a farm that's got 70 to 80% burden of something else.
Mm-hmm. Wow. Okay. So kind of answered the next question, because it was, like obviously the work is still in progress and there's a bit more, a little bit more to discover. Is there anything else that you're hoping to discover? Is there anything that you kind of feel like you're on the cusp of?
There's a lot more, actually. Certainly the thing that we're closest to finishing up is our study on lambs of different ages and how they respond to parasite burdens and what sort of antibody responses they make. So our theory there was New Zealand hasn't ever really thought about this because their lambs are born once a year on pasture in their spring, and they're out on pasture until they're big enough to eat. We have a lot of farms that do rotational lambing systems, accelerated systems, where you might have lambs that potentially have such nice kind of genetic parameters that are born in fall that you might want to keep as replacements that may actually see pasture, but not until the next spring when they're already like 6-7 months old, as opposed to the lambs that are born on pasture in spring and then they're immediately exposed to parasites the minute that they're not drinking from mom anymore. So we took a study where we're comparing one group of lambs that we first started infecting with parasites deliberately, that we knew the numbers, so that we're trying to remove kind of that variation in what you get on pasture, and another group of lambs that were freshly weaned, so that we can see, okay, do the slightly older lambs when we turn them out on pasture have an advantage? Because again, that's going to then play into if you are selling breeding stock from an accelerated system where maybe they're older the first time they're on pasture and exposed to parasites, we might need to adjust what those antibody levels mean in order to fairly identify who is actually the best at making antibody versus a lamb that's being out on pasture right away. So that project is coming to an end sort of late April to early May. So we've had those lambs on study for a year. We're sort of simulating the Canadian season where they've got 5 months on with sort of 3 times a week infections. And then they've got 4 months off over the winter where the parasites are just dormant and they're not on pasture anymore. And then we're going to have another few months on in the spring, just to imagine that they've been now turned back out onto pasture and being exposed again. And every week we've been tracking their antibody levels and their egg output to see what sort of parasite levels are we seeing, but also what sort of antibody levels are we seeing. So not only can we follow what actually happens on a more refined basis than we were able to do out on the farm, like we can't go to a commercial farm every week and sample 400 lambs' saliva. That's just not practical. We can do that on a research basis. So that's the project that we're looking forward to seeing come up, finish relatively soon, have maybe an idea of, hey, what does age do? The next project that's starting up pretty much right on the heels of that one is where we're going to have 3 groups this time, but now we're going to be doing the battle of the sexes. So we're going to have ewes, rams, and wethers, because there is data to indicate that not only is the man cold a thing, but it does also apply to sheep. Sorry, I did that while you were taking a sip of your drink.
Perfect timing. Man cold and parasites. I've never heard those together. So that's amazing.
Yeah. So the thought is that females seem to be better off at making immune responses. And again, that's something we need to know if we're looking at trying to pick the best breeding stock. And so we're throwing a group of wethers in there just for fun to see if, like, because there have been some studies not on parasites where it looks like if you castrate them, it makes them wethers sort of intermediate between males and females. So it sort of feminizes them a bit, makes them a bit better than the intact boys, but not perfect. So we're going to be then following those guys out for a year. And then after that, hopefully we'll be able to then look at different groups where we're comparing the different three kind of big three species of parasites that we have in Ontario, which would be Haemonchus that we've already talked about, Trichostrongylus, colubriformis, which is the brown gutworm, I think it's called, and then Teladrosages circumcincta, which is another sort of stomach worm that they develop, which is relatively common.
Wow.
Excellent. So if we're talking to producers about, what's the most important thing to take from the research that you've done to date, what are your most important findings, that you'd want to communicate to producers?
Probably the most important finding has been one that we've sort of been able to do as a fun sideline, which is tracking just how bad is our benzimidazole drench resistance. So that is things like valbazin, for example. And we have genetic tests on the parasites to see whether they're resistant or not. And it is basically everywhere. So the resistance problem is a huge, huge problem. And so we can't kind of put our heads in the sand and pretend it isn't there because it's brewing, and we really need to try and get ahead of it before we end up in a situation where nobody can actually let their sheep out to graze ever because the parasites are just too bad and are uncontainable. We do have tools we can use to try and help move towards that system. But I like a kind of saying that Laurie Moss used to have. She taught me all about the sort of how to teach sheep producers to do their own fecal egg counts, which is that there are no silver bullets. There are lots of small changes that we have to make to try and make things more sustainable on the long term and keep those drenches working for us. So it's not going to work. Unfortunately, the industry in Canada is too small for us to expect any of the drug companies to come out with a new drench or even to bring us another combination product. And that's ultimately not going to change anything if we continue approaching parasite management the way that we've been doing. So we do need to have a little bit of, I like the term used earlier, Walker, outside of the box thinking. And some of it is actually traditional stuff that happened for centuries before we ever discovered these drenches, like breeding for improved immunity. And then sort of now that we have the tools to understand what in the animal is actually making them more immune, seeing if we can then stimulate those pathways to actually improve the overall animal's ability to tolerate the parasites without us necessarily having to intervene. And then doing some of those management strategies, the classics about making sure you're aware of where your young stock are going and where your old stock have been, and putting your young stock ahead of your old stock because the older animals are going to have had that time to develop a bit of a response. And so they're sort of acting as vacuum cleaners. Maybe even thinking farther outside the box. And if you can have a bit of a mixed operation with some beef cattle don't get infected by the same parasites. So they can act as lawnmowers basically for the parasites out on the grass. So put some cows out, they'll eat up all your sheep parasites, and it will just reduce the number of parasites that are on the grass. And then the sheep will do vice versa for the cattle parasites for the most part. There's a couple of exceptions, but they're not as dangerous as some of the stuff that we're dealing with. So just thinking about some of these other strategies, which are old strategies that perhaps have been overlooked because for so long those drenches worked so well that you could just basically use them to fix any problem. But we can't keep doing that because otherwise we won't have that salvage fix anymore to rely on when we need it.
That's so interesting about the cattle. Okay, so Emma, what projects are your grad students working on this year?
So this kind of touches on a few of the things I talked about, but I can tell you about who's doing what. And so Victoria Alicock is my current master's student, and she is working on the study where we're comparing the lambs of different ages in terms of what they're making in immune responses. So she is coming up to wrapping that project up. My other master's student, Liam Davidson, he's the one that's kind of delving into the Petri dish and trying to understand the cells. So he's been working on isolating what we hope are the specific rare immune cell that has been identified in humans and mice that seems to react to the type of antigen that Carla is. They've never been isolated successfully from sheep before, not because I think that there's any inherent problem, but because nobody has ever bothered to try. So we decided to go in and see. And mammals are mammals are mammals. The nice thing about immune responses is they tend to be very conserved. You tend to be able to extrapolate pretty well from mouse to sheep, from human to sheep, and so on. So he's working on trying to see if those cells exist. And if we can confirm they exist, he's then going to try and grow them. And then we're going to start showing them some of this Carla marker and seeing if they get excited by this. Because if they do, that might be an interesting avenue for us to try and find ways of doing in vitro designs of perhaps some early vaccines involving Carla. This is like years down the line, though. So just don't get too excited about this. It's going to take a while.
Very early.
This is really cool.
Yeah. It's very complicated and it makes my head spin too sometimes, but that's the hope, is if we can try and like figure out some of this stuff. It seems like very high level, but there is always kind of the goal of what's going to happen on farm. And then my new incoming PhD student, Jenny Lynn, she's going to be picking up the next stage of the project, looking at, okay, the battle of the sexes. Are we going to have stronger immune responses in rams or ewes or wethers, or what's going on in there? And hopefully being able to have a look at some of the tissue samples as well to see what the cells on the microscopic level are actually doing in these animals, and then looking at the antibody and parasite levels at the macroscopic level in the animal as well.
Excellent. So your lab has also explored whether the way we store fecal samples has impacted the fecal egg count and larval culture results. Should our on-farm sampling practices change based on your findings?
Yeah, so that was a neat little summer project I did with one of my summer students the other year, Isabella Vitellis. So we found that in short terms, right, we were basically looking at poop samples stored for a period of time we might expect in a vet clinic, right? If you took a sample and you dropped it off with your vet, or if you do your own fecal egg counts on farm, if you took a sample and then maybe waited a couple of days to come back to it. So in that sort of 2 to 3 day range. What was kind of the best way to keep the eggs from doing things that we don't want them to do, right? We want them to stay eggs so we can accurately count how many they are. And what she found was that when you're dealing with short periods of time like that, egg counts are pretty stable if you are storing them in a well-sealed Ziploc bag, or if you are storing them, we basically blended them with water to try and like get all of the oxygen out from where the eggs were, because low oxygen prevents the eggs from hatching. So on the basis of sort of just keeping it as easy and simple and cheap as we can, what we're recommending is kind of the best way to store poop samples to sort of make them as useful for as many possible options in diagnostics as you can is use a good Ziploc bag. Force as much air out of it as you can after you've got your sample to make it as airtight as you can. Doesn't necessarily have to be vacuum packed, but if you manage to sort of like roll them to squeeze the air out, that really helps a lot. And that will be good for at least up to 3 days. You will have steady egg counts, and you just keep them at room temperature. You don't actually have to chill them. And the benefit of keeping them at room temperature is if you decide later on that you would like to know what parasite species you've actually got, that will then keep those eggs that are in that sample completely viable, because we have some species, like I mentioned, Haemonchus is a tropical worm. It does not like the cold. And so if you put those poop samples in a fridge, in some of our other work, we found as little as 2 hours in a fridge can actually start to cause the Haemonchus to just not want to survive and grow. So it's better to keep it at room temperature, because that then is sort of a fair compromise. It's like it's not perfect for anybody, but it's also not going to kill anybody. So you can be fairly confident that if you then later try and figure out which species you've got, that sample after a few days is going to be closer to the species mix that was coming out the back end of the animal when it was fresh.
Huh.
Okay, thank you.
So avoiding the inside out glove. We know it's really convenient, but like let's try and use like nice sealed bags.
That
Right on. Okay.
I have a little side question. So when my understanding with parasites is that it's really hard to poop them out because they obviously are wanting to stay inside. And so, but is it common, as someone who's still new to the industry, but knows a little bit too much about like human parasites? So when the sheep are pooping it out, are you saying it's kind of just guaranteed that the eggs are always going to be coming out at some point? Like they don't have as much of like an attachment as like the live ones?
So this is eggs coming out, because the way that these parasites work is you have the adult parasites live in the animal, and then they're laying the eggs that come out in the poop. And they want the eggs to go out in the poop because those eggs are destined to go to somebody else, to the next host. And so they actually have a period of their life cycle where they live in the poop sample for a little bit, molt a couple of times, grow a little bit, and then they become the infective stage, which then crawls away from the poop into the grass, because sheep are probably not going to want to eat their own poop. So they then climb into the grass, and then they wait for a sheep to come along and eat them. And then they go through a couple of other molt cycles in the sheep before they become adults, and then the cycle repeats.
Gotcha.
So it's that eggs out in the environment part that actually assures the ability for the parasites, after they've been in the host, reproduced, to then transmit their offspring onto the next host.
Okay. So for producers wanting to fine tune their parasite management programs, do you see Parasite ID, like larval culture, being commercially available soon?
Actually, yes. There's a few different kind of initiatives going on. So I believe IDEX, one of the big veterinary companies, has partnered up with a collaborator of ours out in Calgary, John Gilliard, and they are working on basically making like a miniaturized version of what he calls his kind of nemabiome sequencing technique. So what John does out in Calgary is he has a bunch of basically DNA sequences that are specific to the different common species of parasites. And when you send him a sample, he will break it up, take out the DNA sequences, and then stick the primers on, and then amplify it. So make more copies of the DNA so that he can then give you a percentage of, this is how much Haemonchus you've got, this is how much Trichostrongylus, and so on. And so most of the time, people are most interested in Haemonchus because it's so much more pathogenic. It causes so much more disease than any of the other species. So I think IDEX is working on a test that will focus in on the DNA sequences of just the Haemonchus, and then you can send them a sample where, again, they will break up the parasite eggs or larvae or whatever you send them to free up the DNA in those parasites, and then give you about how much percentage of Haemonchus do you have in that sample, which for most farms is going to be useful information for a few reasons, because, again, most pathogenic, but also we have flucovir, closantel, which is a narrow spectrum treatment that specifically targets Haemonchus. So if you know that's mostly what you've got a problem with, you can leave your broad spectrum drugs, because it's then basically making the other species you might have that aren't a problem at the time more resistant to those.
Excellent. So Emma, if somebody is really interested in learning all the things about parasites, they want to read your research results and get all the details, when will the most recent research results be available, and where can they find more information about parasites?
Well, my sheep stuff is going to be a couple of years yet. We're just wrapping up the first of kind of my sheep trials as an independent primary investigator researcher in the coming year. So it probably will take at least another year and a half to potentially 2 years to get those papers out. The problem with field type of research like this is it takes a while. That being said, some of the stuff with Dr. Brad DeWolf and Dr. Andrew Peregrine is actually already out there and published. So the stuff he's been doing with his batch of Ontario sheep farms has already been published in Veterinary Parasitology. I think he's got at least 2 papers out already. So they're out there to hear more about things. But also, like, when we have interim results to share, we're always trying to kind of work with people like Ontario sheep farmers to sort of get the word out there through means like this, through presentations. We have been also working with small ruminant veterinarians of Ontario to try and make sure that they're kind of kept abreast of what the heck is going on. We've presented before at things like the Grey Bruise Farmers Week conference. So we try and get around to some of the producer conferences as well to make it more accessible, because we know it's not always necessarily the easiest thing to go and get hold of some of these papers when we put out the final results.
Amazing. So we'll watch for you at future meetings. And if any of our districts are looking for a guest speaker, you are a fantastic guest speaker. I will attest to that, having heard you speak at district meetings. So we'll put out the word that you are available and a great speaker to anybody who wants to have you come.
Awesome. And my grad students are also great speakers, and it's great for them to get out and sort of meet the people that their work is benefiting, because it sort of makes it more real to them. So it doesn't just have to be me. I know you guys love hearing me yak, but I've got some fantastic students that are super excited about the work that they're doing. So
that's
keep
wonderful.
them in mind too.
Yeah, that's wonderful. That anytime you've got a student who would like to speak, we are ready. We're ready for them. So
Awesome.
We'll get connected with you. And Emma, thanks for joining us so much on the podcast today. We appreciate you sharing all the things about parasites with our listeners.
It's my pleasure again. Like, I love talking about this stuff, and hopefully it's helpful to some people who hear this.
Amazing. And thank you for listening. If you enjoyed this episode of Flock Talk, please subscribe to your podcast app to receive alerts to our future episodes, and please share our podcast with your friends.
For more Ontario sheep farmers content, follow us on social media at Ontario Sheep Farmers on Instagram and Facebook, at Ontario Sheep on X, and visit our website. Can you guess it? OntarioSheep.org.